Hello again, everybody. Mr. Umlar here. Welcome back. In our last video, we talked about mitosis and meiosis, the cell cycle, and how that controls the processes of cells in our body. Today, we're going to go into a little bit of what happens if there are mistakes that happen in that process. And so before we start talking about the mistakes, let's have a quick review, see if I can draw a circle here. Not too bad. And so if you remember, we've got our cell cycle, right, that's progressing along in a cyclic fashion, and it's divided into phases. So we've got our G1 phase, we've got our S phase, we've got our G2 phase, and then at the very end, we've got our M phase, right? And then occasionally we split off, and we have a G0 phase as well. But what's really important about this is that at each phase, there are checkpoints. There is a checkpoint in the S phase as well, but we're focusing mainly on these three right here. And what the G1 and the G2 checkpoints really have in common is that they're checking our DNA. So this G1 checkpoint is just in place to make sure that before we start copying our DNA in the S phase, that there are no mistakes. And then after we've copied, the checkpoint in the G2 phase just makes sure that those two copies we have now are error-free before we go into the mitosis phase or the M phase. And then the checkpoint in the M phase, this happens kind of right in the middle. And what happens there is if we've got our cell with our chromosomes lined up in the middle, the M phase checkpoint just makes sure that our spindle fibers are attached. So whenever all the spindle fibers are attached to the chromosomes, that checkpoint then gets crossed off, and we're able to go on with the rest of mitosis, which would mean that we end up with two cells that each have those three chromosomes in them. And they've all separated evenly, kind of right down the middle. And this is where the biggest mistakes occur in either cell division process. It's something we call non-disjunction. So non-disjunction is when the chromosomes either don't split up at all or they split up incorrectly. So for example, if we're looking at this cell right here that's going through meiosis, we've got our two homologous chromosomes here. We'll call those chromosome 1. And then we've got these two homologous chromosomes we'll call number 2. What you would expect at the end of this process is 4 cells. And each one of them would have a copy of chromosome 1, which is a little bit longer, and a shorter copy, which is chromosome 2. So chromosome 1, chromosome 2, chromosome 1, chromosome 2. And of course, all of these 4 cells would be genetically different from each other because of this is meiosis. In this case, now here, instead of an even splitting of all these, you'll notice that both copies of chromosome 1 and one of the copies of chromosome 2 all ended up on this side, and over here we just have this one lonely copy of chromosome number 2. So what we end up with there is 2 cells that now have extra genetic material and 2 cells here that are missing some genetic material. More often than not, when this happens in our bodies, whether we're talking about egg production or sperm production, those gametes just don't even get used. Our bodies recognize that the DNA didn't split correctly, and those cells are basically reabsorbed into the body, and they never are involved in fertilization. Occasionally, though, those cells make it past our defenses, and they do actually fertilize an egg if it's a sperm cell, or vice versa. Again, in those cases, sometimes the fertilization occurs and the DNA doesn't match up, and the woman's body just immediately rejects the embryo, saying the DNA, there are too many mistakes, this can't develop, and it never develops into a fetus. But occasionally it does get past that point, and then we end up with what are called aneuploidies. An aneuploidy is just a general term for an incorrect number of chromosomes. So that could be too many chromosomes, it could be too few. It's just that the number is incorrect. So for example, the 2 most common forms of that we'll see are trisomy and monosomy, which is trisomy is having 3 copies of a chromosome, so we would be 3N, and monosomy would be 1N, or you just have one copy of a particular chromosome. So in this example, here we've got an egg cell or germ cell going through meiosis, but instead of ending up with 2 chromatids in each cell, we end up with 2 full chromosomes over here and none over on this side. And then down here, we're seeing these eggs get fertilized by a normal sperm cell. So these 2 cells are now monosomic, and these are trisomic over here. These have 3 copies of the chromosome. These have 1. Both of them should have 2 to have the correct number. Again, all of our cells in our body other than gametes are 2N, so we should have 2 copies of every chromosome. There's a very common, not common, but very well-known form of trisomy that many of you have probably heard of, which is trisomy of chromosome number 21, and that's more commonly referred to as Down syndrome. So in Down syndrome, the other 22 chromosomes, there's 2 pairs like there should, or 2 copies like there should be, but that chromosome number 21, there's a 3rd copy instead of just having 2. There's other forms of trisomy out there, but that's the one that most people are familiar with. And then the other form of non-disjunction that can occur is something called translocation. So for translocation, basically what that means is, if we have multiple chromosomes, so let's see if I can draw this here for you. We've got this chromosome here, and then we'll have another chromosome over here. And basically what happens with non-disjunction or with translocation is, let's say this chromosome remained intact, but then our other one over here is now missing this whole arm, and that piece attached itself onto this chromosome. So pieces of chromosomes during this process can actually break off and reattach to another chromosome. And so when our body's trying to figure out what to do with this information, it can't figure it out, because it's like taking chapter 1 of a book and tearing it out and stuffing it right in the middle. It doesn't make sense in that position anymore. And same thing here, our bodies would not know what to do with this information. Sometimes it results in the death of cells. Sometimes it can lead to more serious problems like cancer or some other genetic disorders. On that note, it's worth talking about why cells do die. We've gone through the process of cell cycle, mitosis, meiosis, and one of the main reasons why cells are replaced is because of cell death. But we didn't really talk about why cells might die. And the first reason is what we call necrosis, and this is usually due to some sort of infection or toxin or radiation or things like that that would lead to damage of the cell. And basically what happens here is if these cells are damaged to a point where they can't repair, they will just die, and they'll basically explode and release their contents into the external environment. Extensive necrosis can actually be really bad because it will lead to inflammation and other problems with our immune system. The other form of cell death is what we call apoptosis, and this is also more commonly referred to as programmed cell death. And so sometimes cells they die because they're supposed to. And a great example of this happens during development of fetuses. And as we're all developing, our digits are all fused together. So it's kind of like having penguin hands or something along those lines. And at some point during development, there's a signal sent to those cells in between the fingers for those skin cells to start dying off, and it allows those digits to separate. So in that case, apoptosis, the death of those cells, is a good thing, because otherwise we would be born with fingers that we can't separate, and our hands would not work as well as they're designed to. All right, and the last thing I want to talk about in terms of mistakes being made during cell cycle is how that applies to cancer. We've all heard of cancer. We all know people who have dealt with cancer. And there's a lot of varying causes of cancer. But the hallmarks of cancer are the fact that cells have lost control of the cell cycle, and then these cells have the ability to migrate to other parts of the body. Normally, cells in our body stay where they were formed. They don't break off and go to other places to do jobs in other locations. They're formed in one spot, they're anchored into that spot, and they stay there. So 2 hallmarks of cancer cells is, number 1, they lose control of the cell cycle, so the ability to regulate when to divide and when not to divide, and then the cells gain this ability to migrate or move throughout the body. And as we'll talk about in a moment, that process is what we call metastasis. And so the other thing that's very common, or what we all associate with cancer, is the formation of tumors. There are 2 different types of tumors. The first one, benign tumors, are generally non-cancerous. So a great example of that would be like warts. Warts grow in our skin. They are tumors that stay in one place. They don't harm us really. There's not really a whole lot to be worried about. It's just abnormal tissue growth, as opposed to malignant tumors, and those are the ones that can migrate. Those are the ones that are able to metastasize. And so what makes malignant tumors really harmful is they can travel through our body, they can break off and reestablish in other locations. If you have a tumor that starts in your lungs, that tumor tissue can break off, travel through your bloodstream, end up with your brain, and start growing another tumor there. So these malignant tumors are the ones that tend to spread throughout the body and really cause a lot of damage. So the loss of control is more what we're going to focus on here as it relates to the cell cycle. And there are 2 main types of genes involved here. The first is what we call an oncogene. And these are the genes, if you think about the cell cycle as being kind of like driving a car, the oncogenes would be kind of like the gas pedal. So these are the genes that help the cells progress through the cell cycle. So they'll code for things like growth factor receptors, the signals that cells receive in order to move forward in the cell cycle. The other side of that are tumor suppressors, and these are more like the brakes of the car. So these would control the checkpoints. These are the things that are going to make sure that the cell cycle doesn't progress until we're ready to. And here's 2 really good examples of these things, how mutations in these can lead to cancers. So the first example up here, there's a particular receptor on cells called the HER2 cell, or receptor, and these are specific receptors for growth factors. So they'll receive the signal of a growth factor, and once they receive several of those signals, that triggers a response from the cell to enter into the S phase and the G2 phase and ultimately divide. A mutation in oncogenes can lead to an overproliferation of these HER2 cells. So the cells become much, much more sensitive to growth factors. So growth factors that might not make this cell divide, that same concentration would make this cell divide because it has so many more receptors in it. And that's a common cause of breast cancer. So mutations in this HER2 gene can lead to an overproliferation of these receptors and can lead to uncontrolled cell division. Tumor suppressors would be genes like this that produce something called RB, which is a protein that inhibits the cell cycle. For patients who have cervical cancer, there's oftentimes a mutation in the protein that inactivates these RB proteins, and it basically takes the brakes off of the cell cycle. And so the cell just divides and continues to divide and just skips through all the phases and just keeps going over and over and over through all these different divisions when it really shouldn't be dividing at all. And lastly, last thing we want to talk about is, what are some causes of mutations? And so what are the sorts of things that can lead to these mutations in either oncogenes or tumor suppressors? And some of them you're probably pretty familiar with, so things like radiation, whether we're talking about radiation coming from a nuclear power plant or nuclear bomb or something like that, or the radiation that we're all exposed to every day, which is the radiation that comes from the sun. Smoking is another one that obviously we're all well aware of these days that we shouldn't be smoking. From looking at this picture, these guys up here don't know or didn't know that smoking was very, very bad for them. We now know that this is the effect of smoking on our lungs. So I don't think I need to tell you to avoid smoking at all costs. Other mutagens or other things that can cause mutations in our DNA would be pollutants, whether we're talking about air pollution or water pollution. There's chemicals. Anytime there's a law that exists in every state, so if there's a place that has cancer-causing chemicals on the premises, they always have to post warning signs like this. Some things that have been used in the past that we don't really use anymore would be like asbestos, which is what we have here. But there's lots of different chemicals that can induce mutations in our DNA. And then one that a lot of people don't really associate with mutations or cancer would be viruses. But when you stop and think about what viruses do, they invade our cells, they inject foreign DNA into our cells in order to take over the manufacturing abilities of our cells. That can lead to permanent damage in the DNA. And so there are certain viruses that are associated with cancers. One really common one is the human papilloma or HPV virus, which is a virus that causes genital warts, which is one of the most common STDs that people can contract, mainly because you can have it without even knowing. But this virus has been shown to cause damage to the DNA in cells of the cervix in women and is strongly associated with cervical cancer, which is why a lot of doctors are now recommending that young ladies get the HPV vaccine. But anyhow, that's pretty much it for the topic of what mistakes can be made during meiosis or mitosis. In our next unit, when we get to DNA and how it replicates and is used to make proteins, we'll come back to this a little bit more. But that's it for today. So hope that was helpful.